we realize so actually there is the ground run going on at the same time. So these here get it completely wrong all the time, and it's actually great to see that actually few people turn up. The ground run is the biggest events that we have monthly, and not only that, but it's got a much better catering. So it's much more appealing to people. So today I'm glad to be here because these clinical seminars we try to get going, and someone has been converted from the ground round to the clinical seminars. I'm sure there'll be more people coming in. Okay, so let's start talking about asymmetry brain connections. And the reason why we're talking about asymmetry brain connections is for two reasons. Well, first of all, asymmetry, it was a kind of a topic that was very interesting among psychologists and cognitive neuroscientists, very much alive in the 1980s, and not so much now. But it was very interesting because we thought that by studying asymmetry, we could actually discover something about consciousness and specialization of functions. So that is a topic that is coming back, actually. And the reason why it's coming back is because we have a better neuroimaging methods. We haven't really moved far in terms of psychology, but in terms of neuroimaging and electrophysiology, we're now tackling the problem. So that's why this kind of a field is re-emerging and will become more and more popular in the next few decades. Now, when we talk about connections, I'm afraid, again, half of my slides are historical because I'm afraid I had to fill this talk because this talk, asymmetry of connection, there's not much experimental evidence out there. So I will kindly introduce you to the anatomy of the connection, etc., and then tell you what is there out there in terms of the anatomy of connections. Okay. So, why asymmetry? Symmetry and asymmetry. So, one of the features of the human body is actually the symmetry. If you observe, we have more converted people. Were you at the ground round that you decided that? Oh no. Okay. So, right. Okay. So. One of the features of the human body, I mean, actually, the animal system is the symmetry. So we have 2 legs, 2 arms, 2 eyes, etc. And even when we kind of see some organs like the heart, which seems asymmetrical, it actually comes from the fusion of 2 arteries, which are perfectly symmetrical. So while symmetry seems to be logical. From an evolutionary point of view, it makes sense. If you lose one kidney, your survival probability of surviving are higher if you have another kidney on the other side that could continue do the same functions. But what about asymmetry? What is the advantage of losing one side and remaining only with one organ? And of course, we not only we find ourselves symmetricals or asymmetric, but also we're surrounded by objects around us which are symmetrical or asymmetricals. And just like it's by poor chance. This is a glass of Guinness, and that today we probably celebrate. That's for you, Sean. St. Patrick's Day. So when it comes to connections, what we're talking about? We're talking about mainly 3 types of connections. We're talking about association pathways. In here, they are colored in greens. These association pathways at different areas within the same hemispheres. They have usually a longitudinal course. Then you have a contour pathways that connect one side to the other, and these are colored in red. And then you have a projections pathways, come from the cortex to the cortical structures and vice versa. So ascending and descending. This is the classification of white matter tracts. So today I'm going to talk to you mainly about, you will see projection pathways, because that's where all started, and talk you through association, and finally like a few slides on, 1 or 2 slides on commissural pathways and asymmetry. So now you have a broad category. You have association pathway, commissural pathways, projections pathways. Let's have a look a bit more what kind of tracts are there. For the commissural pathways, you have the biggest corpus callosum connecting the majority of the cerebral cortical areas, except the temporal lobe, which is connected by the anterior commissures. Please come in. That's okay. Then we have another 2. Please come in. Okay. Then we have projection pathways. The main projections pathway, of course, is the internal capsule that contains a lot of ascending and descending pathways, but also the fornix. The fornix that connects amygdala hippocampus to the mammillary bodies. Then we have the third group, the association tracts. There are the uncinate, arcuate, cingulate, inferior frontooccipital fasciculus, inferior longitudinal fasciculus. So these are the tracts that belong to the 3 different categories. If you have any questions, please stop me each time you want to ask something. So let's start with the projection systems. We know that the projection system connects cortical areas to cortical structures, and that is pretty much bilateral. We have 2 cortical spinal tracts, one on the left and one on the right. The projection system changes its name according to where you are in the brain. So on the top is called corona radiata, simply because on a sagittal images you can see it's like a crown, okay, a radiating crown. That's why it's called corona radiata. But when it enters into the internal capsule, it's called internal capsule fiber. But the same fibers really. And then they take different names according to where they end, or according to the structure they run through. Now, how long who discovered the projection systems, and what are the major milestones? One of the Vesalius is really in the Renaissance. So he was able to kind of look at these white matter tracts, the projection system. He actually described the internal capsule, but he couldn't really understand what it was made of, because he could only see white matter. He couldn't really see fibers. It's really when Thomas Willis started using a new method for doing dissections, which consists in extracting the brain from the skull. So you can see the difference between these 2 dissections. In the Vesalius representation, the brain is in situ, still there inside the skull. For Thomas Willis, he was able to take it out and start looking at these the brain and other structures, subcortical structures. And that's by doing dissections of these subcortical structures that he discovered a striatum. And he attributed a function to this striatum, which is a motor function. So it's really Thomas Willis, long time ago, that identified this structure. Now, he knew that it was made of fibers, but really it took a long time to understand how these fibers actually converge from the cortex into the internal capsule. And really, this is what we know nowadays about one structure of the internal capsule, which is called the cortical spinal tract. Now, the cortical spinal tract is the tracts that we use to control movements, okay? And it originates from a mainly precentral cortex. So you've got a central sulcus. The precentral cortex is right in front of the central sulcus, belongs to the frontal lobes. And that's why you have all the neurons that control different muscles in the body. And you can see this is kind of a homunculus. These are somatotopic distributions of these neurons. Now these neurons are quite long. Some of them are shorter because they have to stop at the level of the brainstem, but some others continue all the way down to the spinal cord. Now, what really happens, they rotate in the brain so that the phase neurons, the phase axons are anterior and the legs are posterior. But what really is important is that at the level of the pyramid, or just below, fibers cross from one side to the other. Okay? And this crossing is called decussations of pyramids, or decussation. Now, the decussation was initially discovered for the first time by François Porphyry-Petit, a Frenchman. At the same time, it was discovered by an Italian, Domenico Mistichelli, but we don't have the picture of Domenico Mistichelli. Somehow, French were much more, they were better at having self-portraits. They were more aware of their scientific legacies. And most of the Italians, I mean, they may discover, but they didn't really make picture portraits of themselves. So there you have. Michelle, you'll be happy about that. Yeah. Okay, so what is important? Because it's really here, for the first time, that we have the beginning of this kind of a branch, this discipline of starting looking at asymmetry of white matter connections. Okay, so that was 18th century, but we have to wait. So Willis described for the first time decussation. We have to wait really another few decades before the first true experimental evidence of an asymmetry of the cortical spinal tracts becomes evident. And it was really Paul-Emile Flechsig, who was a great anatomist, not a great clinician, but great anatomist, who used to stain developing brains, so fetuses and newborns. And he was able to look at the myelination of these fibers. But during this work, not only he discovered the areas that are myelinated very early in life, that he called primary sensory motor areas, and the ones that myelinate later, he called it association areas. Okay, but he kind of came up for the first time with evidence that this decussation occurs in between 75 and 90% of the normal populations. So in other words, the 10% of you, between 10 and 25% of us actually, that control mainly the right hand with the right brain, and the left hand with the left hand, the left hand with the left brain. But the majority of us, up to 90%, control the left hand with the right brain. So we have a decussation, okay, and the other way around. Not only that, he realized that when these fibers cross, so 9 out of 10 cross of us, but only 40% of us have an asymmetrical crossing. And it usually is the fibers that from the left go to on the right, they are bigger in volumes as compared to the ones from the right to the left. Okay? This is the first difficult, I think, passage. Should I repeat this? Yeah? Okay, so one of us, 1 in 10 of us, doesn't have crossing. 9 out of 10 have a crossing fibers. Of these 9 out of 10, almost 50% have a crossing which is asymmetrical. So his fibers from the left that cross to the right are bigger compared to the ones on the right that cross to the left. Okay? Yes, please.
Bigger means bigger, that means a different amount of
Well, okay, I'll go. You're right. I mean, he couldn't measure volume. So what he was thinking of, what he experimental evidence was that actually myelinate first, are more myelinated. So, with myelination you will see later that comes with volume increase, but for now, you're actually right, it's actually myelinate first, probably. That's what he wanted to say. So, the fibers, 50% of us that are asymmetrical, have these myelinations that occurs more for the fibers going from the left to the right, while the fibers that go from the right to the left myelinate later. Okay?
So did you say that 1 in 10 people don't have the crossing fibers?
Yes.
But does that mean their left and right brains are rotor representations of them?
I couldn't really tell you about that.
Right brain the other way around?
It's difficult to say because then you have to take into account handedness and a lot of things, and I couldn't really tell you about that. But let me finish this kind of a first line of evidence, and then we can think about that, because this is amazing evidence that comes from 100 years ago, but you'll see at the end, nobody really pursued it. I mean, it stayed there, but it's so interesting. So that was the first post-mortem study. What happened after that? Well, we had to kind of wait another probably 40 or 50 years for Americans Yakovlev and Raki to confirm those data, but also to tell us that when these fibers cross from one side to another, the fibers that cross from the left to the right cross first. They are at the top of the decussations, while the fibers that cross from the right to the left, they're at the bottom of the decussation. Right? So, and that is in 90% of us. I wouldn't be able to tell you whether there's one that cross from left to right, that are at the top, also have an asymmetrical pattern of distribution. Nobody really look at that. So what comes to your mind when someone is telling you, well, actually, some of us, half of us, have this asymmetry, and actually the fibers that go from the left to the right either decussate first, the top, or actually they demyelinate first during life. And so what comes to your mind,
Are there any consequences of that? And is there any simple way of determining which group you belong to without resorting to MRI or cortical stimulation?
This decussation. Well,
Not inside, that's easily done.
Yes.
Is it a simple test for determining which category?
Yeah, you might think that because this is the cortical spinal tract, is there assessment of handedness and manuality skills. But actually, when people are trying to see that, didn't correlate. There's this study done by Kertész and Geschwin. They studied how this kind of asymmetry correlate with handedness. It was not a lot, of subjects, but they couldn't see any difference in left-handed versus right-handed. Not only that, but when Nathan and Smith and Deacon look at how big is the spinal cord, so in this case we are talking about after the decussation, down into the spinal cord, how this asymmetry, they found that actually on the right side, 75% of cases of the spinal cord were bigger on the right side as compared to the left, but they said, well, that's 75%. We know that right-handed is much more than that, so this cannot be the correlate of handedness. So it's a kind of a puzzling issue, this one, because we have clearly an asymmetry for some of the characteristics, but this doesn't really correlate with handedness. Actually, I have to tell you about this volume, this more recent one, 2001, published in Brain. They took 10 brains, they sliced and they stained the fibers, and they look at the part which is mainly the cortical spinal tract, which is mainly running in the cerebral hemisphere, so above the decussations. And they found, again, a volumetric asymmetry, but again, in 70% of the brain. So the left side being bigger than the right in 7 out of 10 of the brain they dissected. So stay with me for a while. Things get complicated, and then I'm trying to summarize to give you kind of a final message. So when we looked at asymmetry the studies that I told to you are about postmortem studies, and so far you have, if you include Flexig, then you include Rademacher, and the other two, you have 4 studies in total. Not much. Considering that cortical spinal tracts, it was kind of a discovery a long time ago. But when you're trying to look at this in vivo evidence, and you try to separate the cortical spinal tract for the rest of the projection systems, so overall the projection system doesn't really differ between left and right. So all the fibers, the thalamic fiber, the cerebellar fibers, there's not really much different between the left and the right. But what we found is that actually the number of streamlines, which is a surrogate for volume, is lateralized for the cortical spinal tract, and again is bigger on the left as compared to the right. So this is a kind of a nice in vivo evidence that supports, not all the way around, postmortem support the in vivo evidence for a lateralization of the cortical spinal tract. This is is interesting because it looks like, if you look at the thalamic, so the red ones are mainly are ascending fibers. They bring information to the brain. And the cortical spinal tract is mainly the ascending fibers that we use to move muscles, but also to change environments around us. So it looks like the information that we receive from the environment is come from both sides, but somehow we selected one side of the body to act, to respond, to react to the environment, which is, for me, from an evolutionary point of view, is very, very interesting. Now, so this is my summary slide. So if you got lost at some point during the previous talks, I'll try to summarize. And this is really pretty much all the hard evidence that we have for asymmetry of connections. So above the decussations, the fibers are coming out from the cortex into the internal capsule. 70% of the population has the left side, the left cortical spinal tract is bigger on the left compared to the right. Another 17% of us has no asymmetry on both sides. And there's only 12% of us that actually has the left cortical spinal tract above the decussations, which is bigger as compared to the right. Now, when it comes to crossing here, again, so between 75 and 90% of us have the crossing going this way. The 10% we're referring to have only the red fibers. Okay? Among those that have this crossing, 40% are asymmetrical, like in this case, where you have left fibers going from one side, from left to right, are bigger than the fibers going from right to left, or myelinate early at birth. And 90%, again, of us have this kind of a left fibers going to the right, being more dorsal than the fiber from the right going to the left. On the spinal cord, this is the Nathan studies in 1995. On the spinal cord, the right spinal cord is bigger than compared to the left in 75% of the cases. Sean?
In those who don't have crossing fibers, is there still an asymmetry between two sides, two tracts? So like,
Yeah.
they just have the right and the left and they don't cross.
Yeah. It's difficult to say. I mean, these studies were done on small samples, and I think I didn't go back to the original source, also because it's in German, so I couldn't read in German. But looking at Steffi, who's is my German brain, so it would be worth looking at that. But I don't think there is much evidence for that. I mean, this is a kind of what I'm trying to highlight here is that areas that have not been explored and that probably in anatomy of the 21st century need to be explored and to tackle with modern neuroimaging methods. I couldn't really tell you about the brain, the functional anatomic organization of the brains that don't cross. Right, so any questions about this? But I think this kind of is a nice evidence that for decussations and a spinal cord, there's good evidence that actually there is an asymmetry. Yeah?
Just on those studies, what is the percentage of handedness, left and right handedness?
Okay, only one study looked at left-handed, which is the Curtis and Gershwin 1971. There was an archives of neurology. It was a postmortem study, and they had a few left-handed, a handful of left-handed though.
Okay.
And in those left-handed, there's no difference in terms of asymmetry of crossing.
On the symmetry of crossing.
Asymmetry of crossing. Or the decussation. So for example, what they look at is this decussation here. So this left from right is dorsal, more dorsal than the right from left. This is more ventral, okay? Can you see that? That is a difference, okay? This, you can find its anatomical features in 90% of the right-handed, and the same percentage comes out in the left-handed.
In terms of volume, yes. Is there a correlation?
I don't think they have done volume. It's almost impossible to do volume unless you use the modern methods that used by Rademacher, but they couldn't do volume. I think they could count the number of fibers, they could kind of look at myelination.
Well, my question is, do you think that those findings, not to dorsal ventral or the symmetry of crossing, do you think those findings are mostly because when you use a side more than other to white matter responds plastically and becomes bigger?
Well, that is a kind of egg and chicken question, isn't it? I mean, if you don't use your muscle, you lose muscles. So there is a correlation between muscle and exercise, but you don't know, which one comes first. So if you have motor neuron disease, you exercise less, but if you're actually lazy, then you lose muscle. So which one comes first? I couldn't really tell you this. My opinion about this is that actually cortical spinal tract symmetry is not the neuroanatomical correlate for handedness. I think we should look into other things, because handedness is not about strength. It's about coordination, dexterity, but skillful is the movement of fingers. I think that is to do a lot with the cerebellum. But let's, and then we will need a study to do that. But let's not even talk about that. So that is a projection system. That's all I have to tell you about projection system. It's not very much, but it's the evidence that I can find by writing this chapter for a book that is coming out this year on brain asymmetry. Now, let's move on to the association tracts. These are very important. Association tracts are the neuroanatomical basis of cognitions and behavior. And I'll take you back to 200 years, back in Germany, where John Christian Ryle and Frederick Butzer discovered and described most of these association tracts. And they describe it, and they use a particular book that used Latin names that remains almost unchanged nowadays. So, very solid anatomical work. When these association tracts came along and were discovered, people quite rightly saw that these tracts were important for higher cognitive functions like language, for example, or reading, or praxis, movements of hands and fingers. And people like Wernicke, Lichtheim, and Desjardins, they came up with network models of cognitive functions. Okay, so they didn't think about an area, two areas, but actually they thought about networks that were important for carrying out a specific function. Like, for example, this Wernicke model language, B is an area in the frontal lobe for speech articulations, and this A is an area in the temporal lobe for auditory comprehension to language. And he thought about a network that connects these two areas. Now, they knew that these functions were specialized on one side, but apparently they kind of thinking around the symmetry was not as finely tuned as they're thinking about other things. So, for example, Wernicke, when he was talking about language, he knew that language was a function on the left hemisphere, and but he represented the language network on the right hemisphere. This is the original figure of Wernicke. So, as you can see, this is clearly our right hemisphere. So it's a huge mistake. So he was really kind of distracted when he was talking about asymmetry. Now, behind that, what is really important, they all assumed that there's these language pathways or reading pathways on the left hemispheres. But when it comes to explain why there are these networks on the left, they went back to cortical localizationism. So they all assumed, for example, Wernicke assumed there is an area on the left hemisphere that's specialized for auditory comprehension. Desjardins, the same, an area in the angular gyrus that is specialized for reading. So on one hand, they were very much into networks. On the other hand, they were much into, when it comes to specialization of function, they were cortical localizationists. The guy who came up with the first model about lateralization of cognitive function based on lateralization of the connections was actually Lippmann. Lippmann was a student of Wernicke, and he thought that for praxis is a normal movement, coordinate movement of arms and etc., limbs. He thought that there is a network that somehow connects primary sensory areas to primary motor area, and that these kind of connections are weaker on one side, in particular the connection that goes to the right hemisphere as compared to the connection that goes to the left hemisphere. So when it comes to explaining why you have apraxia only for lesion to the left hemisphere, but quite rarely when it comes to lesion to the right hemisphere, he thought, well, the reason is not because there is a center dedicated to praxis on the left hemispheres, but because the connections going to this left side are stronger as compared to the right. So this is a kind of the first real model of dominance of cognitive functions based on lateralization of pathways. So when it comes to hard evidence, postmortem evidence, or lateralizations of association tracts, I couldn't find a single study. There's only one study probably on the uncinate done by Tim Crow that's kind of a asymmetry. But for the most obvious tracts, like for example the arcuate, there is no post-mortem evidence whatsoever. That's quite surprising, isn't it? So if you're thinking about a future line of research, I think this is a good one because it's been so neglected for many years. So this is a study that we did with Michel. So when it comes to association tracts and look at the left and right, is there anything that is asymmetrical? Well, the first thing is, this is the obvious arcuate fasciculus. The arcuate fasciculus is a language pathways on the left. So, and this is quite lateralized to the left, both in males and females. But when it comes to other tracts, like, for example, the cingulum or these ventral systems, quite surprisingly, what is it? The number of streamlines. Okay, I think this is the other way around. Sorry, guys. I apologize. The number of streamlines should be here. So this is kind of volume, okay? So just forget about this is wrong. Okay, the number of streamlines, the volume of the arcuate fasciculus is lateralized to the left. But this is not the only tract that is lateralized. The other tract that is lateralized is the inferior longitudinal fasciculus. There are other tracts, and we'll see later other tracts as well. Now, so let's focus on the arcuate fasciculus. A few years ago, when we looked at the arcuate fasciculus, we found out that actually there are 3 segments. How many of you have heard of or seen this slide before? Because if everyone is, I won't really go through. Okay, not many. So I'll do it for you, and be patient if you've heard these 10,000 times. But basically, the idea is that Wernicke's and Broca's areas on the left hemispheres are connected through the red fibers, which are direct connections, but also indirect fibers that pass through the yellow segment, which is the posterior segment, and the anterior segment, which is the green segment. When you start looking at lateralization of the long segment, okay, you have that 60% of us in here don't have direct connections between temporal and frontal areas on the right hemisphere. So 60% of us are extremely left lateralized. Another 20% of us have some fibers, red fibers to the right, but remain left lateralized. So the left long segment is much bigger than compared to the right. Finally, another 20% is bilateral and symmetrical. Sometimes more on the right compared to the left, but never left more than the right. Okay, so this is kind of an evidence here that actually there is an asymmetry for a subcomponent, at least a subcomponent of this perusylvian, the arcuate fasciculus. And then when it comes to then gender differences, you'll see that 85% of males are extremely left lateralized. So we belong to the group 1. Only 15% of us have a bilateral distribution, either bilateral but still left lateralized, or bilateral and symmetrical. And the females is 14, 30, and 30. So there's a kind of a degree overlap, but they are more bilateral represented. Now, when it comes to correlate this asymmetry with neuropsychological performances, on the California Verbal Learning Test, you will see this trend, which is this correlation, which is significant for both genders, that where the group 1, the ones that are extremely left lateralized, perform worse within the normal limits, but perform worse on this test as compared to the ones that are bilateral and symmetrical, and the ones that bilateral but still left lateralized have intermediate values. So there's a kind of a here for this test, the California Verbal Learning Test, which is a test that you do by, is a list of 16 words that you have to remember, objects. They are linked together by meaning, by semantic associations. So the better you remember how to link them together, the better are your performances. Now in this test, for this test, it's more advantageous to be bilateral than left lateralized. So is not an advantage, being extremely left lateralized, for this task that requires semantic clustering and verbal memory. I couldn't really tell you about other language tests because we didn't really test language or visual spatial functions, but for the most obvious language function, like verbal fluency, for example, there was no correlation whatsoever. Now, why this is important? So, we've seen that we are lateralized not only for cortical spinal tract, but also for language pathways. And what is interesting is that this lateralization changes across the lifespan. So, for some of the tracts. So, for the long segment that we've seen is quite left lateralized in the majority of us, before adolescence, the left side is biggest compared to the right, and remain left lateralized throughout adolescence and early adulthood. Okay? For the anterior segment, there is a kind of an opposite situation. Differences are not huge between left and right, but before adolescence is the right bigger than the left, and when it comes to adolescence, that asymmetry remains the same. As if these two components of the language pathways, the anterior segment and the long segment, lateralize quite early in life, and they don't change during life. But there's a component here, the posterior segment connects temporal to parietal, that starts off being right lateralized, being bigger on the right compared to the left, and then when adolescence kicks in, then it becomes bilateral and reduces in volume in both sides. So this is telling you that probably anatomy changes, especially in critical periods of life like adolescence, and that this is not true for all the tracts. For example, in this case, it's only true for the posterior segment connecting temporal to parietal lobes. Now, this is important from an evolutionary point of view. So this lateralization, well, is a kind of difficult to say because, for example, one of the major features that happen during evolutions, that as a humans we increase the number, the volume of connections between temporal and parietal lobes. So this kind of a posterior segment seems to be quite a significant features of the human brain. When it comes to other tracts like the anterior segment, this is a kind of it hasn't changed that much during evolution. So it may have some relevance to explain the development of higher cognitive functions in humans. But what I think is interesting is how to study lateralization during brain development as a way to look at how the brain heals himself or recover from some injuries. And these are preterms. This is a preterm man, actually. He was 20 years old when we scanned him, and he was born very, very preterm. We're talking about his weight at birth was less than 1 kilogram. Okay, so, and he's kind of, so that was, and there was 26 weeks. You know, he stayed in utero only for 26 weeks, which is, at the limits of any survival. So this kind of a white matter damage that you have in preterms, you have a lot of white matter pathology, and in this case there was a kind of this atrophy of white matter, this increase in volume in the lateral ventricles. That wasn't particularly true for the left hemisphere as compared to the right. Is any psychologist here? Any psychologists of the? Yeah, there's Michelle. So, what would you expect in terms of language functions on this subject that has virtually not left hemisphere? Sorry? Yeah, or should it have some kind of language deficit. That's why we do clinical anatomical correlation. I wouldn't like to have this brain. I don't know you, but probably have a worse brain, but I wouldn't like to have this brain, put it that way. And yet, we test him, and his language functions were better than mine. Right? So, even if you test me in Italian, which is my language, so not because of a language barrier, but even if you test, he had a very good language skills. So how is it possible? That's the fight. That is a kind of against any clinical anatomical correlation rule. Well, we did the dissection of the arcuate fasciculus on his brain. We couldn't find any arcuate fasciculus whatsoever on the left hemisphere. This guy had no arcuate fasciculus on the left. And yet he was speaking to us. In fact, he had a 3-segment, which is a pattern that we see more commonly on the left. He had only the 3-segment only on the right hemisphere. Somehow, this guy with virtual no arcuate fasciculus on the left managed to shift all his function to the right. And again, we're going back to their questions, whether are the functions that reshape the anatomy, or the anatomy that explain the function, I couldn't tell you. But this guy, what's interesting is actually this cortical spinal tract. You remember the left being bigger on compared to the right, etc. His left cortical spinal tract was very thin, and they had a quite thick cortical spinal tract, and he was left-handed, without any family history of left-handedness, as if again he developed he relied on the tract, the cortical spinal tract, that had less alterations. Now I'll conclude. This is towards the end with the commissure pathway. So you might think, oh well, how can I study the commissure, being the commissure just right in the middle, going from left to right and from right to left? How can I study the anatomy? Well, we have some evidence, psychological evidence, and probably Michelle knows better about this, that when you present something on one and some visual or auditory stimuli on one side, and it goes on the contralateral side, and then you have to kind of shift everything on the other side, it looks like that it's easier for some information to go from the right to the left as compared the information to go from the left to the right. There's something there. Is it the Pfaffenberg paradigm? Is anyone familiar with this? I'm not psychologist, not even neuropsychologist. Well, believe me, there is a kind of a physiological evidence that it's easier to shift, so to send across the corpus callosum some information from right to left as compared to left from right. And the corpus callosum was really, really a topic that was very important to study lateralization of main cognitive functions. It started off with the dissecting of the corpus callosum in patients with epilepsy, and ended up really with a Nobel Prize to Roger Sperry for his studies of people with split brain. Now, when it comes to the corpus callosum, you can use tractography again. You can look at the different segments of the corpus callosum, but also you can look at the cortical connectivity. So you seed on one side of the brain and try to track and to see where the fibers or the streamlines are going on from one side to the other side. And by doing that, this is kind of a recent paper that came out by Gazzanica's group. So when you try to seed from one side of the Brodmann area 5, for example, when you seed it on the left hemisphere, and you try to track across the corpus callosum, you have much less fibers than when you try to go from the right to the left. As if, and this is particularly significant for these areas 18 and 19, which are not the primary visual areas, they are not calcarine cortex, but are kind of associative visual areas. So in other words, and these studies have been interpreted as if there is a kind of a asymmetry of connectivity between left and right. So much easier, much more connections going from right to left as compared from the ones going from left to right. Now, experts on DTI tractography, like Flavio Dell'Acqua, would think this is all rubbish. And may be true, but there are so many problems. First of all, when you track from right to left, and you track and trying to reconstruct the fiber, it doesn't really mean that those fibers are going from right to left. It may well be that actually those fibers are coming from the other side, but as if you are kind of navigating the river, on the other, going upwards. So this is the first thing. The second thing is actually to go from one side to another, you have to cross a lot of structures. So if you have a bigger arcuate fasciculus, for example, on one side, or a cortical spinal tract is big on one side as compared to the other, it's more likely that your trajectory are hindered by obstacles. So it's nothing to do with actually callosal connections, but with something else. But I think we need a study that would prove that. But until we have that, we have to take this as kind of a preliminary evidence for this. And so Flavio is developing this technique. As you can see, those guys have used a method that's called DTI tractography that allows you to dissect out the corpus callosum. But when you use another method, which is the convolution tractography, you'll see that actually there are much more fibers than we thought. And so I think those studies need to be confirmed, and we need to go back and do the same probably experiment by using the spherical convolution. Because when you use DTI tractography, you have a problem of crossing. So when it comes to crossing, you can't visualize the fibers. So this is again another area that's pretty much not been explored and needs to be explored. So what are the future directions for in this kind of huge topic which covers asymmetry of brain connection? Well, first of all, we know that some tract exists, like the superior longitudinal fasciculus connecting frontal to parietal fibers, but we are not able to visualize it in the living human brain because the DTI, the diffusion tensor model, is a kind of is limited by these crossing fibers. So the development of new method like spherical convolution would allow us to look at these tracks that we know exist, but we know very little about. So this is the first line of research. The second is actually, okay, I have a slide for this. So this is the SLF as described in monkeys. So far, if you take all the anatomical illustrations of the human brain from postmortem dissection, they all show you the arcuate fasciculus or the inferior longitudinal fasciculus, but there are not really descriptions of the superior longitudinal fasciculus in the human brain. Okay? And for the first time, this is the work of Flanders and Michel. They used spherical convolution to show actually that this three-system SLF1 exists in humans, and are pretty much similar. So Michel is now studying how these systems lateralize. And one of the predictions is, because this SLF is very important for visuospatial processing, that somehow is lateralized on the opposite side, on the right hemisphere, because underlies many visuospatial functions which are lateralized to the right. So that's the first thing. The second thing is about looking for those tracks that we don't know whether they exist. And so how is it possible to look for tracks that we don't know they exist? Well, because, for example, there are so many fibers that connect neighboring gyri. And we know that these fibers exist because they were described by Sachs. I think it's Henry Sachs? Henry Sachs, okay. Which, for the occipital lobe, he had this project of coming up with an atlas of these short fibers for each lobe, and he managed to do only the occipital lobe. But for the occipital lobe, we have the detailed descriptions of these short fibers, which are within each lobe. So they're called intralobar fibers. They don't leave the single lobes. So we know they exist for the occipital lobe, and they may well exist also for other lobes, like the parietal, the frontal, the temporal, but yet they've not been described. So again, if you're thinking about a line of research, just try to describe these tracts, and you may see, you know, your name at some point linked to one of the tracts that you discovered. And finally, I think it's what are the correlations with behavior. We've seen that the correlation with behavior and lateralization is it leads to unexpected finding, like the California Verbal Learning Test. I think it is important to start and try to understand how this evidence of lateralizations could explain differences in behavioral performances using multimodal imaging, and trying finally to translate this novel evidence about asymmetry to the clinical arena. That's what we're doing with Steffi, trying to see whether there's 40% of us that has some kind of language pathways on the right are the ones that recover better after 6 months after a left-sided stroke and then having aphasia. We have an ongoing trial at the moment that's trying to use exactly that. I think that was my final slide, probably. Oh yeah, and thank you for listening, and I wish you have a nice St. Patrick's Day. Any questions that you have about asymmetry? There probably was a full injection of everything. So next time we will, I think probably is going to be in April, the next talk. And have you received the email about these seminars? And so you got the email, yes? So I will probably send it by email or post it the next talk. Okay, thank you.